These researchers and veterinarians are on their way to assess the health of a moose who is showing a lack of coordination, weakness, and paralysis. They will sedate the moose and run some tests to determine if it should be euthanized. But what is exactly that the moose is suffering from? Some background information regarding moose are that they are placental mammals classified within phylum Eutheria and clade Artiodactyla, or even-toed herbivores with hooves called ungulates. Reports of moose in Minnesota can be traced back to as early as the 1900s. Moose hunting was culturally significant to the Native American tribes and early settlers who moved to the area for lodging and mining. While moose hunting seasons occurred periodically throughout the 1900s and early 2000s, the state of Minnesota closed moose hunting to all non-tribal hunters due to the significant drops in their populations. Necropsies of moose have revealed many environmental and pathological stressors that have led to their abundant mortality. But their periodic declines are often correlated with higher than usual populations of white-tailed deer. As a preferred host for brainworms, white-tailed deer rarely suffer symptoms from this worm. However, severe brain and spinal cord damage can arise in moose when they accidentally consume brainworms. When moose forage on grass, the intermediate host, an infected gastropod, can be inadvertently consumed by the moose. The parasitic larvae will make their way up to the spinal cord and brain of the moose. The brainworm will destroy nervous tissue, causing inflammation and begin to manipulate the neurological behavior of the moose. An adult brainworm within the brain or spinal cord of the moose can be fatal, causing paralysis, lack of fear, malnutrition, and starvation.
University of Minnesota postdoctoral researcher Dr. Mark Dittmer was part of a multidisciplinary research team that worked to understand the environmental factors in seasons when moose in northeastern Minnesota are most susceptible to brainworm infections. Outfitting 173 adult moose with a GPS collar programmed to record moose location every 1.25 to 4.25 hours allowed researchers to assess their movement patterns. The collar would transmit a mortality signal if a moose was stationary for 6 hours, allowing a team of investigators to perform field or lab necropsies to determine the cause of death. The researchers used a statistical model of brainworm infection in moose, regardless of the cause of death, as a function of several explanatory variables. The researchers assessed relationships between brainworm infection and moose exposure to factors of infection risk that were based on space use by season, land cover types, deer density, environmental conditions, and demographics. Results indicated brainworm infections were present in 24 of the 173 moose, with 5 necropsies concluding that brainworm was the primary cause of mortality. The open diamonds represent dead moose that were infected with brainworm.
Results indicated that infection with brainworm during the summer season were weakly associated with wet soil and land that had been developed for human use. Moose with autumn home ranges that included more shrubs and conifers that were closer to Lake Superior and individuals associated with higher proportions of wet environments regardless of season had increased infection risk. The spring model showed the strongest relationship between exposure and infection, potentially due to moose foraging on ground vegetation during spring. As expected, higher proportions of mixed and coniferous forests within spring home ranges resulted in reduced risks of infection. However, the probability of infection for moose was not influenced by deer density, although deer densities did not vary greatly within the study area. This highlights the importance of also considering both moose space and environmental conditions in understanding infection risk.
Many studies and management strategies continue to focus on white-tailed deer population density in order to control brainworm infections. However, environmental conditions and habitat can play a significant role in the risk of brainworm infection. Future moose management plans should focus on reducing the frequency of contact between moose and high-risk environments, in addition to limiting the density of deer in order to help preserve a stable moose population. Given the small scope of this study, a larger-scale study on the frequency of contact between moose and high-risk environments should be done to further solidify or modify its findings and to gain more insight into the complex life cycle of brainworm. Moose are the primary large prey species of a boreal ecosystem like those found in northern Minnesota. If moose in Minnesota's boreal forest were to disappear, the loss of their role in changing vegetation composition would be felt across the entire ecosystem. Moreover, the geographic expansion of brainworm and deer has important implications for several other northern species, including caribou, elk, and moose. Further research to improve understanding of moose disease ecology can help determine what steps can be taken to reduce their susceptibility to brainworms and other harmful parasites. The models used to investigate the disease ecology of moose and brainworm could also be adapted to fit other disease ecologies. Understanding the life cycle of brainworms and moose can help in understanding the life cycles of parasitic organisms in other animals and even humans.